Set
I
the
am.
Okay.
Yep, let's go.
All right. Hello, everyone.
Hello.
Good morning, afternoon, evening. Gotta get all the time zones. Good to see you all. I'll get started in just a minute. All right. Welcome everyone to webinar number 12 with the International Oak Society and Global Conservation Consortium for Oak. I'm Amy Byrne. I'm based at the Morton Arboretum, helping coordinate the GCCO and also on the board of iOS as well. These webinars are co-facilitated with myself and Roderick Cameron, who is our GCCO coordinator for Western Eurasia, North Africa region, and also a member of the iOS board. And I'm just showing my screen here, which we're just wanting to thank our generous support of our institutional members. So just highlighting this here, as always, the beginning of our webinars. Just to run through the agenda, we have some announcements. I'll pass off to Roderick in just a minute. And then following the announcements, we have two presentations in store today. Hugo Afonso is presenting on flower development and diversity in Quercus. We'll break for some Q&A for Hugo, and then we'll do a short poll, actually, or short survey that we'll launch in Zoom. And then we'll move on to the second presentation, Greg Tyler, who will be presenting on root cuttings of evergreen oaks. Again, some time for Q&A, and then we'll wrap up with some discussion if there's any just general questions or comments about iOS in general or feedback for future webinars. And then we'll go ahead and wrap up. So that's what we have in store for us today. Thank you again to our presenters who are joining us and sharing their work with us. Roderick, I'll pass it off to you for announcements.
Great, thank you, Amy. And actually, the first thing I need to say is to thank Amy Byrne for hosting these webinars. She's done every single one so far and done a great job. So huge thank you to Amy for doing that. There's not a whole lot to announce on iOS, just a run through our events. We just held two very enjoyable Oak Open Days in the UK. Perhaps some of you will have participated in those. They were a visit to White House Farm in the UK. That's Maurice Foster's garden, which was fantastic. I mean, difficult to focus only on the oaks because there's so many botanical treasures there. And the next day we were at Winkham Arboretum, completely different. It's a wild arboretum, or a wilder, but some really interesting oaks there, and also a very interesting management of the woodland. So I did get the impression that people were pleased with these events, a lot to learn and a lot to enjoy. Coming up, we have a tour of Turkey. You'll see that on our website. There's still places available, so if you're interested in joining, please do check this out and make sure you register, because places the tour may fill up. 7-day event, traveling through Turkey, looking at oaks and also other plants and other events of cultural interest. And then in October, our tour of Southeast USA. I hope very soon to share the full itinerary, so those who have reserved can book their hotels. But there's more info on our website, and I'll share the link in the chat in a minute. So I think that's all I have on our events. We are busy planning next year's events, but we will announce those later once we have finalized details. Okay, I think that's all from me, Amy. So back to you, and I think we can get started on the presentations.
Yeah, great. Thank you so much, Roderick. So yeah, we'll pass it off to our first presenter, Hugo. So whenever you're ready to share your screen, go ahead.
So it should be sharing, hopefully.
Yep, we see it.
Wonderful. So hello, everyone. I'm Hugo. I come from the University of Minho in Braga, Portugal. I'm a PhD student, um, focusing my work, of course, in the development of flowers in Quercus, also in Biopolis, CIBIO from the University of Porto. I'm a bit in both places. So flowers are, of course, one of the greatest innovations in land plants. They are largely responsible for their huge evolutionary success, which ultimately led to a huge variety of floral shapes, sizes, colors, morphologies, fragrances, and everything in between that we're all so familiar with. And the majority of these flowers fits within the canonical hermaphroditic model. Essentially, these flowers are mostly formed of four consecutive whorls of sepals, petals, stamens, and pistils. The stamens, of course, being responsible for the formation of pollen, and pistils being pollinated, conducting that pollen towards the ovary, which will further on develop the seed and the fruit. However, for evolution, a more specialized form has emerged in these kinds of flowers, with the complete separation of the male and the female organs in separate structures. In here, we already have our example from the Quercus, in which male flowers only have a whorl of stamens and a whorl of sepal-like tepals. And female flowers only have pistils at the center, tepals around that center, and a cupule of concentric scales that will enlarge and develop the acorn cap that we're all also so familiar with. And yes, the male flowers are arranged in unisexual catkins, which typically emerge from the bottom of the new branches right upon bud bursts in the beginning of the season. These inflorescences are usually very elongated and pendant, which facilitates the pollination via the wind. The female flowers will emerge further towards the apex of the branch in the axils of the new leaves. And there is usually a temporal separation in the emergence of male and female flowers that can range from 1 to 2 weeks, for instance, in Quercus robur, up to 2 months in the case of Quercus sube. During my work, I'm trying to focus on as many oak species as I can in Portugal. But most of my work has been focused on Quercus cantabrica. Quercus cantabrica belongs to the Quercus robur group. It is native to northwestern Iberian Peninsula. It's a deciduous Marcescent species. And the taxonomic history of this species has been particularly complex, at times a little bit confusing. Essentially, it was previously considered a subspecies of Quercus robur broteruana, and a recent work by Carlos Villalobos and collaborators has shown that there is enough distinguishing, both in terms of morphology and in terms of genetics, to considering it a proper individual species. I invite you to read the recent species spotlights, in which Roderick and Carlos go a bit further on the taxonomic complexity of the species. Regarding flowering, the species has exactly as I mentioned before for the wider genus, sorry, with the male flowers emerging at the bottom of the branch, the female flowers emerging further up. Quercus cantabrica, in similarity to Quercus robur and other pedunculate oaks, has the particularity of having, as the name suggests, a much longer peduncle, rather than being sessile or only having a few centimeters, as in the majority of species. In this species, it can reach up to 16, sometimes 20 centimeters in length. The flowers themselves fit within what we would expect. I want in here to highlight the center of the male flower that does not have any parent organs when it develops normally. And this becomes relevant when we take a closer look, not at what is normal, but what may not be normal. During my work, I have found a wide variety of floral anomalies. In male flowers, I have found both what appears to be a normal stamen, but much smaller. You can see the size difference between the one on the center and the ones around it. And sometimes, instead of a stamen, there appeared to be a poorly developed pistil without a proper genoecium, and that did not appear to display any sort of pollen receptivity. And all of these flowers were eventually shed normally as normal male flowers. In addition, I found some inflorescences that appear to be normal female inflorescences emerging from the axil of the new leaves and sometimes above other completely normal female inflorescences. However, these ones, rather than having female flowers, they have what appears to be normal male flowers, but of course in the number of flowers that the normal inflorescence would have. So this shows us that the development of the flower itself, the identity of the flower, whether it is male or female, appears to be independent from the identity of the inflorescence itself, meaning the inflorescence developed as female, but the flower developed as male. But perhaps a more striking phenotype that we have found in our search was the occurrence of fully complete hermaphroditic flowers. These were flowers that at the first glance appeared female with pistils and the cupule, but on a closer look they displayed fully developed stamens capable of producing pollen. And we did try to germinate this pollen, and the pollen was capable of germinating. And the majority of flowers were pollinated, developed acorn, and the acorn matured at the end of the season, which means that these flowers are 100% viable, 100% hermaphroditic, rather than being just morphological, as was the case of the previous flowers that I've shown. I also want to highlight the fact that these stamens are clearly separated from the center whorl. So you have the center normal whorl of pistils, and around it you have the stamens developing. In here I have removed the scales in order to make it more obvious. But that was not always the case, and it means that sometimes in this unusual whorl that shouldn't be present, some pistils develop instead of stamens. And in some rare occasions, but still a few times, some stamens were found to coexist with some pistils in this third whorl, in this middle whorl, that should not exist, which once again reinforces that the development of the organ is not 100% dependent on the position of the organ, because in this third whorl, it can be either a stamen or a pistil. Hermaphroditic flowering has been reported in several species, particularly in Mexican white oaks, but it was not reported so far in Quercus and other European white oaks. So we're trying we're working on a manuscript to make these results available in a proper publication. However, to have a flower, we need to develop it. And oaks usually oscillate between periods of active growth and periods of dormancy. At least the temperate species. The tropical species tend to skip this latter step. So they have dormancy, they experience bud bursts. As I mentioned, the flowers emerge at the bottom, and the female flowers emerge further up towards the top of the branch. But this is not where the development stops. The majority of oaks usually have several vegetative flushes over the same season. The majority of them has at least 2 or 3, depending on how long the season lasts. Essentially, the buds that were formed in the same year will themselves experience bursts and develop a new branch. Again, this is observed in the majority of species, and in the case of Quercus seedlings that were grown in controlled conditions, a maximum at least of 13 consecutive flushes has been reported. So for as long as you have good enough conditions, the majority of oaks will continue to grow successive flushes. What is not normal, however, is for these flushes to be accompanied by another flowering event. It has been reported as well in several species, but it is usually considered a rare event. But most likely a rare event that is brought upon by atypical conditions, which unfortunately we are experiencing more and more, including here in Portugal. And in the last 3 years, we have seen occurrence of secondary flowering, both in summer, throughout summer, and throughout autumn, in the secondary flush, and sometimes in tertiary flush as well. And what is interesting is that the flowering can occur throughout the entire season and not just at a specific point, like the initial flowering that occurs in the first bud burst. And this flowering sometimes has normal morphology, but sometimes not. The flowering that emerges immediately upon the second flush usually develops normally as a female inflorescence with female flowers, and of course some hermaphroditic flowers that somehow appear to be more numerous in this scenario than in the first flowering, which already shows at least a certain degree of divergence. However, further ahead in the season, these flowers here are both from summer, the inflorescences do not develop normally. They develop almost as an intermediate state between male and female inflorescences. Essentially, they have a large amount of flowers, similarly to the male inflorescences, but they are much stiffer, much thicker, and pendant, in similarity to the female flowers in inflorescence. These flowers were usually, the majority of them, very anomalous. They had either a large amount of stamens or the organs were in different proportions. But some were also found in the same inflorescence to be female or male, with the majority of them in between to be hermaphroditic. The flowers that displayed gynoecium developed the beginning of a fruit, but all of them were shed before the end of the season. So this flowering was essentially wasted, and it means that the plant was not able to produce acorns out of it. This is something that, as I said, is not it has been reported for other species, and we have found here in Braga in at least also Quercus ilex, the whole oak. In this case, there is also a very strong deviation from what is typically normal. The original flowering, the quote, well, normal flowering, let's say, has 2 or 3 flowers, and the stalk is only a few centimeters wide, few centimeters long. But summer flowering in this species was also found to be much longer. In this case, it reached over 20 centimeters and displayed a huge amount of flowers. In this inflorescence in particular, it had over 30 flowers, and with the added bonus that the majority of them were also hermaphroditic flowers. Again, it has also been reported in other species, and particularly for Quercus ilex has been reported, especially the hermaphrodite, but not to such a wide degree. So it is again reported as rare and not in the entire canopy of the tree. And we have found it in the entire canopy of the trees that we have been observing. In Quercus suber, the cork oak, however, it becomes a little bit more interesting because Quercus suber, depending on the region and depending on the tree, can display either annual or biannual acorns that develop in the same season as they are born or in the following season. And some trees are able, under certain circumstances, to create both in the same season. And what we have found here in Braga is that a few trees display summer flowering in, again, late summer, with apparently normal male, female flowers and a few hermaphroditic flowers as well, in inflorescences that are also much longer than what is typical. The difference is that rather than beginning growth right away, they became dormant shortly after, and they remain dormant but alive during the winter, rather than being shed like in Quercus aurocantaurica and Quercus ilex. And these flowers resumed growth in the following season, and they were able to bear acorn at the same time as acorns from the following season that were normal annual acorns. And so it appears that at least this species can take advantage of this secondary flowering to increase its reproductive output and increase the production of acorn. And so this phenomenon in other species that cannot have biannual acorns most likely is a remnant trait that is being, again, brought back by atypical conditions. However, because of evolution throughout the passage of time, they lost the ability to maintain these flowers and to properly develop them. I also invite you to read the post from last year in Alan Coombs' blog that also reports some very similar phenomenon in both Quercus ilex, Quercus obtusata, and Quercus altapista. Another interesting detail that I have found in these inflorescences is that some of them appear to have what appear to be a gradient of male identity, with the flowers towards the apex having much more stamens, and the stamens much larger than flowers on the bottom. And flowers here in the bottom sometimes did not have flowers at all. And I did mention that sometimes we have both male and female flowers in these inflorescences. The male flowers appeared at the apex, the female flowers appeared at the bottom. If we take a step back and look at other familiar species, the Castanea genus, chestnuts, is very close to oaks. However, it differs in the arrangement of the flowers. The flowers are arranged in male catkins and bisexual catkins that have female flowers at the bottom, at the base, and male flowers in the remainder of the inflorescence. And this is something that is also seen in other species like Lithocarpus. And it is widely considered to be essentially an evolutionary stepstone between the development of unisexual flowers and the unisexual catkins from a hermaphroditic ancestor. And during this transition, there was also the transition in pollination type, because chestnuts are pollinated by insects, while the majority of oaks is pollinated by the wind, because the separation of inflorescences and dependent male catkins favors that type of pollination. However, when we see these atypical inflorescences that appear to be a step back in terms of development, it makes us wonder whether the pollination type in these inflorescences would be the same, because even though they are also pendant, at least the female flowers are again not arranged in the same way, and because the morphology becomes more similar to this, states, it is possible that they also favor entomological pollination rather than anemophilous. In addition, considering that in the formation of these atypical inflorescences appear to be sometimes a bit random, because you did not find always male flowers when you have these inflorescences, the presence of stamens in some otherwise normal female flowers may be a way to ensure pollination when these flowers emerge, but there is no male inflorescence to pollinate them. Great question that we have in our lab is exactly what is coordinating the separation, what is coordinating the separation of female flowers from male flowers, and why they grow the way they grow, in order to try to figure out why sometimes they don't, why they sometimes grow an intermediate state. Not only in morphological point of view, but also genetic point of view. And in terms of genetics, this has been extensively studied in hermaphroditic model species. Essentially, this model, the ABCDE model, it postulates that different combinations of genes will lead to the formation of different organs. Each gene essentially works as a molecular switch that will activate a wide variety of downstream genes. And those genes, in turn, will orchestrate the formation of the organs. And so, different combinations will lead to different outcomes. If we combine A and E, we have sepals. But if we add the B class to the other two, we have petals. Exchange A through C, you have stamens. Remove the B, and you have carpels. And so it is. It's already easy to speculate that if you change the arrangement and the expression of these genes, you can both change the identity of the organ, but also restrict that identity to a certain structure. This is a very elegant model. It has been applied to a wide variety of species. And if you remove some of the genes, which has already been done in the model species, for instance, removing the B class gene will lead to the conversion of petals into sepals and stamens into carpels, as has been shown in mutants that are lacking at least one B class gene. In here, I want to highlight the pistillate gene. This gene appears to be more central, if we can say it, for stamen development, because when we remove it, rather than having a conversion of stamens into carpels, which also happens in some cases, but rather than having a straight up conversion, we sometimes have complete absence of the third world. So we have those four worlds, sepals, petals, stamens, and carpels. The stamen world simply disappears, and the other two are converted. If, however, we remove the C class genes instead, which of course are orchestrating the reproductive development, the stamens become petals and the carpels become sepals, as is represented here by the gametes mutant. And so in our lab, we have taken this model and tried to apply it to oaks. Not only oaks, we also tried it in Castanea sativa, the European chestnut, but also, of course, in Quercus suber. And the results that we found were roughly what we would expect. The B class genes that are involved in stamen development are much more expressed in male inflorescences and male flowers. But crucially, the pistillate gene that I just mentioned before, the Quercus homologue was not found whatsoever in the female flowers, which once again hints at a major role of this gene in the initiation of the organ, rather than just the development of the organ itself. And when we took this gene and we inserted it in the pistillate mutant that I showed before in Arabidopsis, the gene removed from Quercus was able to restore the stamens, which means that the gene in Quercus does hold the ability to initiate the stamens in that species, although here is obviously applied in a different one. So we also explored this dynamic in Quercus sativa, as I mentioned. In here, the dynamic was largely the same, with the exception that the piscillata gene was found, albeit to a low level, in the female flowers. And when we take a closer look at the female flowers, underneath the sepals, we have some small underdeveloped staminode, which is not considered an anomaly. It's considered widespread in the Castanea genus, and does further hint at the involvement of piscillata homologs in the development of staminoid structures. And so, during my work, I've been trying to expand this research to Quercus cantabrica. And so I analyzed these same genes in various stages of flower development, both male, female, but also in hermaphroditic flowers that we previously haven't found in the other two species. Of course, I'm trying to focus specifically in the development of the stamens in otherwise normal female flowers. And so, trying to break this down, essentially, again, the dynamic was much the same. The B-class genes were more expressed in the male flowers than in the female hermaphroditic flowers. In here, once again, I want to highlight the piscillata gene. Similar to Quercus suber, it was not found in female flowers, but it is present in hermaphroditic flowers, which, of course, once again, it makes a lot of sense because its presence is once again correlated with the development of the stamen. And in addition, right here, it is not present in hermaphroditic flowers in the second stage. If we take a closer look at the second stage, these flowers already have senescent dry stamens that already release the pollen and are functionally dead. And so this indicates that the expression of the gene is restricted to the stamens and not widespread in the remainder of the flower. Regarding the C-class genes here in the bottom, once again here agamas, but also the shatterproof gene, agamas appears to be the only one that is present in both flowers, while shatterproof is not present in male flowers. And this, to us, was a bit striking because when we analyzed it in Quercus suber, it was found in both flowers, both male and female flowers. And our hypothesis at first was that it was involved in the development of both stamens and carpels, and that agamas was having a much less important role. However, the absence of shatterproof here in Quercus cantabrica male flowers indicates that that agamas may be sufficient to create the stamens, and that shatterproof may have one of two dynamics. Either the function has changed in the evolution from between Quercus suber and Quercus cantabrica, meaning it holds different functions in both species, or the function is the same, but the expression in male flowers may not be relevant, meaning it might be expressed but might not be having an effect. However, it is very peculiar considering that it is a central gene to the development of flowers, but the flowers themselves are exactly the same in terms of morphology, both in architecture, both in the number of stamens and the development of sepals. So even though the genes are much different, the shape is the same. It is something that we are currently trying to investigate as to why exactly that is. Currently, my focus will be to try to expand this knowledge to other species, try to see where is the line that separates one thing over the other, and try to take a closer look at the involvement of these genes, both in the development of similar flowers in other species, but also different flowers in the same species. I would very much enjoy trying to associate these genes with the various morphologies that I have shown you before. Of course, I can't end this work without thanking everyone that contributed to it, particularly these 3 students, which were PhD students before I have arrived. All my lab mates for keeping me sane for all of this work, the Foundation for Science and Technology in Portugal for our funding, and all of you for your kind attention. Thank you.
Wonderful. Thank you so much, Hugo. Great presentation. All right, now we have time for questions. Feel free to unmute or put it in the chat. One question in the chat, Hugo, do you observe this also in other sections?
Yes, both. I have found it in Quercus mesinifolia in the botanical garden in Porto, in Quercus suber, so Secnus sever, I believe, in Quercus ilex. So already three different groups just in here, but we have found it in other species as well. Secondary flowering is something that we have seen. I couldn't find the appropriate photos, but we found it also in Quercus Virginia, and it has been reported in all continents in a wide variety of locations. And it's particularly interesting because in here it's supposedly not normal, but if we take tropical species into consideration, tropical species, of course, have to have multiple flowerings. If they do not have dormancy, it's normal that they will have it successively.
Okay, thank you. Just a few comments and conversation going on in the chat, so feel free to browse that at any point, Hugo. But Roderick, go ahead.
Yeah, Hugo, that was fascinating. I'm still trying to recover. That was a lot of information and really interesting. Now, correct me if I'm wrong, but this is not the question of hermaphroditic flowers has not really been discussed before. I recall, I think it was mentioned there's a Californian species, could be Quercus parvula, and someone could correct me, that someone reported, oh, wow, we found male and female flowers together in one plant. So if this is becoming more current, is there a theory as to why it's happening? Is it perhaps climate change that is provoking this behavior to be restored? You mentioned just now that it was also found in tropical flowers, so perhaps that's part of it. Are there any theories about that?
Maybe both the hermaphroditic flowering and the atypical inflorescences. It has been reported in several species, but it's widely indeed it has been not very discussed. I have here a few examples. We have Quercus tumosa, agrifolia, John Tuckery, Durata. In the Americas, we have a few in Southeast Asia, the Cyclolanopsis, generally Flora, Nevius, Stanisi. It appears to not be isolated. And even in the same species, in different locations, has been found. So it seems to be something, and our hypothesis is that it is not something that is rare, but something that is largely overlooked. That is probably just hiding beneath our noses, because it took a while until we found it ourselves. It was only when we went to underneath the magnifying lens that we actually could find it. And now that I already have a trained eye, I can find it at the naked eye. But it is usually not easy, especially in species that have a lot of trichomes. Because, for instance, Quercus pyrenica, the trichomes themselves hide can hide the organ. In the case of pyrenica, we haven't found it yet, but as an example. But yes, our suspicion is a little bit of both. It may be uncommon, but not rare. But atypical environmental conditions may be bringing it back, essentially, and making it more frequent.
Great, thank you. Thanks.
Thanks, Roderick. Brian?
Yeah, I guess I have a bit of a two-part question. The first part I think is simple, but could you remind me, so is the typical situation in primary flowering in oaks, the male flowers come out first, the catkins release their pollen and do their thing before the female flowers become available? And then the second part is, what do you think about, what are your thoughts on self-incompatibility in oaks?
Yes, in the majority of cases, the male flowers come first. They begin pollination before the female flowers have receptivity in the same individual, but across the population, there's of course a huge overlap. In Quercus cantabrica in particular, I have found a lot of overlap in the same individual. So there is there is a lot of potential for self-pollination. I'm not certain about the prevention of selfing. I know that in some cases flowers abort when they have their own pollen, but our suspicion is that it is not 100%, especially in the secondary inflorescences, because the only pollen it could have had is from itself. So most likely there may be some self-incompatibility, but that incompatibility may also be being disrupted. There are some cases, I don't remember the species, but there are some cases in which they come upside down, let's say. The female inflorescences emerge before the male freshness. But it is rare. Most often it is male freshness that emerge first. Most likely, that switch may be in different points of the tree.
Okay, fascinating. Thank you.
Great. Thanks, Brian. Any other questions? All right. So again, there were a number of comments and discussion in the chat, Hugo, so feel free to read through those when you get a chance. Thanks so much, everyone, for your questions and for the discussion. And thank you again so much, Hugo, for this amazing presentation. I learned a lot, so thank you for sharing. All right, we will move on to actually I have a short poll or like survey, 3 questions. Roderick and I are just curious to learn more about where participants are joining us from. So let me go back and relaunch it.
While you prepare that, Amy, I forgot to say at the beginning, this is the first time we've opened up our webinars to the public. So we're very delighted to welcome any non-members to this event. We hope you enjoy it, and we hope you stay in touch and learn more about a society. And yes, part of in the poll, if you scroll down, you'll see all the questions. We also want to know if you're a member or not and how you heard about this event. Everyone sees that you can scroll down to questions 2 and 3, right?
Yeah, people are answering them. So I'll just give
Great.
it a couple minutes for people to answer those questions.
Super.
There was a question in the chat, Roderick, on getting CEUs for ISA for this.
We have no experience with this. We'd have to check with Ryan, our president. I know he's good at this, and we do it for our conferences. We have not done it for our webinars as yet, but we would have to look into it to see if we can do it for the future.
Yeah, good question. And yes, hopefully in future webinars we can add other answers or responses for where people heard about this event. Thank you, Dirk, for mentioning that.
Yes, that's a good point, Dirk. Actually, I was trying to set up the third question just for non-members only, but we couldn't do it in the poll. So if you're an iOS member, I'm sure you probably heard this through the QPU, through our e-bulletin. But it's interesting to see how non-members heard about it.
All right.
Okay.
Final call. And then we do webinars quarterly. There was a question about that in the chat. Right,
Yes.
Roderick? Quarterly?
That's right. Yeah.
Okay.
Roughly quarterly. We
Yeah.
announce them on our website and through our communications.
All right. Well, I'll leave it open while our next presenter, Greg, if you want to queue up your presentation, and then once it's up and ready to go, I will end this. Great. We see your screen. I think you want to start maybe on the first slide.
Yep.
Okay.
What is happening here? All right, there. You seeing that?
Great.
All
Yes. Looks great. Thank you.
good? All right. Well, thank you everyone for coming to hear me talk about this. And thanks for inviting me, John Lazinski. I think I pronounced your last name correctly. If any of you saw the post on Instagram for the talk I was going to give, John gave it a really, I like the title, but it might have been a bit misleading. It was called How to Root Evergreen Oaks, which sounded like I was going to tell you how to change a flat tire on your car. And it would be a set of few steps and it was all going to work very easily. So I think this is a good title, Improved Methods for Rooting Evergreen Oaks. It's still very challenging, obviously, but I feel like I've come up with some methods where I've been more successful than average. But if you also saw a little bit about me, you saw that I was a calculus teacher. I've taught college for a long time, but I'm currently teaching at a high school. But plants are just a hobby for me. I really got into collecting cacti and succulents when I was in graduate school, getting a degree in physics. I've also been a physics teacher a lot, and kind of continued collecting cacti and succulents until I finally bought a house here in Portland, Oregon. And so I had a yard, and then I got into woody plants, particularly the Mediterranean type plants that grow well here in Portland, like such as manzanitas. And of course, we need the evergreen oak from California. So I really got interested in woody plants, and living here in Portland, I got to meet Sean Hogan of Cistus Nursery, who has a huge selection of rare plants from all different groups of plants. But he's especially fond of oaks too. So that's where virtually all of my plants come from, is from him. Initially, when I really got into oaks, Sean had some really rare individuals that he only had one of, large trees, and I wanted to be able to propagate them. So initially, I tried doing tissue culture, and not only was it very hard and time consuming, it was also just really a complete failure. I never had any success doing it. My explants were always getting infected. So I didn't do that for terribly long. And then I decided to try to just get better at just rooting cuttings the old fashioned way on a misting bench. Sean Hogan has done that with quite a few different species himself. But I was wanting to try to just get better at it and try to use some of the methods I learned in tissue culture, largely just dealing with sterilizing as much as possible, preventing infection in the cutting. Because when I first began to try rooting cuttings, what I noticed is that the first thing that would happen is that the base of the stem would turn black very quickly and begin to move upwards. And I'd observed that in tissue culture also. So I thought one of the keys to trying to get cuttings to root more effectively was to prevent fungal infections in the cutting, which I'm assuming it's fungal infections, or it could be viruses too. So start moving through. One thing I realized I forgot to do is label the plants here. So I'm going to tell you what they are. A lot of these are Quercus pinnavinia. This was one of the trees that Sean Hogan had gotten seedlings of probably about 12 years ago, where he got acorns of it and started to grow them up. And initially we thought it was Quercus tarahumara, but then he had some trees that flowered. And as soon as they flowered, when produced female flowers, we could tell that it was not tarahumara, but looked more like urbanii. But then, being at the iOS conference in Oaxaca this past fall, talking to some of the experts there, we determined that it's actually this northern population of urbanii that now has its own name, which is called Quercus pinnavinia. So you'll see quite a few pictures of it, because it's probably one of my favorite oaks to grow, and it turns out to be relatively easy to root too. I've rooted quite a few cuttings of it. So here's two more pictures of the same Quercus pinnavinia. So first off, just kind of go through the steps of when I typically take cuttings and kind of what the protocol is that I use. I've typically had the best success basically taking cuttings right about now, just when the first flush of growth is beginning to harden off. The leaves at the terminal end of the stem may still be a little bit soft, but I usually wait till those are getting pretty firm and then take the cutting. Though I did take some cuttings this past weekend of some very young new growth just to see what was going to happen. So I'm hoping that as the summer goes on and I am continuing to do more experiments now, that maybe I'll put a short article on the iOS site with some of the findings that I had from this summer of taking cuttings of different ages. Both of these are also Quercus pinnavinia, and actually this one down here is a rooted cutting. I rooted this cutting last summer, and those leaves you see there, the green leaves, are the leaves that grew on last summer. And then this is the flush of growth coming on in the greenhouse this year. Now, I did have some success taking cuttings in the fall this past year. I actually took them not too long after I got back from the iOS conference in Oaxaca. So that would have been late October, and actually had a bit of success this fall, though it does take longer doing it then. It really kind of took virtually the entirety of winter for the roots to come on from the ones that I took in the fall. Whereas when I take these cuttings right around now, best case scenario, I will have roots growing on a cutting within about 2 weeks. Sometimes it can take as much as a month. I've also had some cuttings that I've had stuck under mist for a year before they eventually start to produce roots. So you never know what's going to happen. So this is actually the picture on the left. It's the exact same stem as this, just taken a few weeks later. And then this is Quercus resinosa, another one that this one is in the yard. It's not terribly hardy here. It's frozen down to its stump 2 times, which is actually, well, it's a bit unfortunate because I want it to get big, but it probably never will. But when it does freeze down to the stump, it will sprout up 50 shoots from the stump, and those cuttings end up being relatively easy to root. So typically the younger the plant is, the better. I've actually rooted several cuttings from seedlings that sprouted this past fall. So if you have a species that's you only have a few seeds of, that you really want more of, as within the first year of that plant growing, if you begin to take cuttings of it, those are I'm not going to say extremely easy to root, but compared to a mature tree, they're way, way easier. Because I would say with a large mature tree, I've had almost 0 success rooting cuttings. So the younger the plant, the better. And if you have a tree that you don't mind cutting down to a stump, it isn't terribly large and you're willing to make that sacrifice, cutting it down to just above ground level and allowing it to produce shoots, you'll probably have great success rooting those cuttings. So this is the one thing which I think is maybe will improve your success if you're into trying this, is using a systemic fungicide on the cuttings. And this is kind of something that I've decided to do as a result of doing tissue culture, because that's something you have to be very, very careful about with tissue culture. So I'm going to try not to just read through this, but I have a lot of details here because I assume you'll have access. Hopefully we'll be able to have access to these slides if you want to kind of follow this protocol. So I'll first get all the cuttings, and you can see here's a picture. This is the day before I'm going to actually put them in a rooting hormone. So I've already trimmed the leaves if I need to. For smaller leaf species like the one here, and actually this is an unidentified species. Sean Hogan has a lot of oaks on the property that we do not know what they actually are, but other than the fact that they're from Mexico. And so this is one of them here. And so the really small leaved ones, I don't trim the leaves at all, but larger leaved ones, I'll trim them back so that they're all, roughly, you can see about the size in inches, a square inch, or maybe about 5, 6, 7 square centimeters in surface area. And so here I have them stuck in this pre-treatment, and you can see what the mixture is here. So the solution I mix is a liter of water. So in a liter of water, we would put roughly 500 milligrams of vitamin C, and then 1.5 to 2 milliliters of this systemic fungicide. This fungicide here is it's a phosphorus-based salt, and it is a systemic fungicide, but because it's a salt, you can only use so much of it. And so if you read instructions on this, you're advised to never go above 1 milliliter, 1% concentration. So basically what I'm recommending here is going up to 2% concentration. And the reason I was able to eventually increase the concentration had to do with the conversation I had at the iOS conference in Oaxaca with a young man that does tissue culture with woody plants. And he said, try adding a bit of vitamin C to the solution. It may allow you to add more of the systemic fungicide without the leaves just turning brown and falling off the cutting. And indeed, that has shown to be true. I can come close to doubling the amount of the systemic fungicide by adding vitamin C. And it's pretty amazing to me. I'm not a chemist. I'm a calculus teacher. So actually, because 500 milligrams is not very much added to a liter of water. Probably if you drank it, you would barely be able to tell that there was any vitamin C in there. It might be slightly tangy. But it seems to have a great effect on the cutting, preventing the leaves from turning brown. Indeed, these are ones that I stuck almost a week ago, and I was just looking at them yesterday, and some of these have literally no browning on the edges of the leaves, and I did a 2% solution, basically putting the upper limit of it. one thing you will find that I've found is that some species are much more sensitive to the salt in this systemic fungicide than others. So I have learned that specific ones that I've been trying to root that I've not been successful with yet are much more sensitive to it. And so I will go down to about 1.5% solution for those. But many of them you can go up to 2% and the leaves look perfectly fine. And hopefully this base of the stem does not turn brown. It does not get infected. But one other thing I'm not sure if I mentioned a second ago is that when I put these into this pre-treatment, I've cut the base of the stem at a pretty sharp angle, but I leave the stem a bit longer than I want it to be, because after it's gone through this pre-treatment, I like to cut off a good bit of the base of the stem before putting it into the rooting hormone. My assumption being that right after I've cut it off the tree, during that time period of it being cut off the tree and this fungicide getting into the stem, that there's the potential that some infection has begun and that it's slowly, I don't know how quickly this would occur, but it is slowly traveling up the base of the stem. So after this treatment with the fungicide, I like to be able to cut off as much of the stem as possible. You can't always do that. Sometimes you're taking a cutting and it's not very long, so you don't really have a whole lot to work with. But as much as possible, I like to cut off a lot of that base before dipping it into the hormone. All right. So here's a nice little, so this is one that just rooted recently. So this is a Quercus crassifolia, and this is from a seedling that grew last fall. So this plant grew indoors under artificial light just for a couple of months in the fall. And then I moved it outdoors into cooler temperature, shorter photo period, so it can have a bit of dormancy. And then this spring in the greenhouse, it put up this really vigorous flush of new growth. It was actually so vigorous that it was kind of flopping around, and I really don't like when trees do that. So I just chopped off that flush of growth and allow it to sprout up from beneath, and it's actually already done that. And this occurred within 2 weeks, so I was really excited about that. So let's go into the hormone part. So I use a very high, what you would consider a pretty high concentration of IBA. I just buy powdered IBA. It's actually some that I still have from doing tissue culture. So you just buy this it's in powder form if you've never used it, and you want to buy 100% IBA. It's pretty expensive if you buy a little bottle of it. It is not very cheap, but I mean, it lasts a pretty long time. And I use it as a 1% solution by weight. And to do that, you dissolve it in 50% ethanol. So if you work in a lab doing tissue culture or something like that, you probably have access to 50% ethanol. I don't. So the easiest thing to do is just to buy vodka, because it is essentially just ethanol and water for the most part. And you really do have to get 100 proof vodka for it to be 50% ethanol. Initially, I was using, most vodka is 80 proof, so it's 40% ethanol. And what I found is that if the IBA was at 1% concentration and 40% ethanol, after dipping a few cuttings in it and little particles would get in there, the IBA would precipitate onto those particles, and then you get all these crystals forming, and then you have to warm it up to dissolve it again. But 50% ethanol, it never precipitates out. So you just want to be sure to do that. So basically 1% solution by weight, I just measure out 100 grams of the 50% ethanol and then add a gram of IBA. And of course, you can scale that up as much as you want it to be. So as I said, the first thing I do is I want to cut off as much as possible of the original base of the stem from the cutting. Maybe I try to go up at least an inch if I can, about 3 centimeters from the original cut. And so you can see that here. There's the second cut that I made, this diagonal cut here. And then what I do is using a scalpel. I use like a surgical scalpel, not a razor blade. A lot of people like to use razor blades, but to do this, really have to use a scalpel because you have to be very precise. And you can just see it here. I make a vertical cut. You can see right where this top root is coming out. That's where I did the cut. And I cut. So for this stem, this is a crassifolia. It's not a really thick stem. I'm maybe going to cut no more than 1 millimeter deep into the surface. And for this one, I cut from here down, looks like to here. I believe that's the base of the cut. And so for a thin stem like this one, I'll just do 2. So I would have done 1 on this side and then 1 on the other side that you can't see. For really thick stems like Quercus pinnavinia, I'll maybe do 3 or 4 of those incisions around the stem. And this does seem to have an effect because, as you can see, these roots are coming right out of that cut. And I've had many cuttings where an infection has actually started at the base of the stem, and I will see basically the base of the stem will start to turn blackish brown, and it will be moving up, but that IBA has gotten into this incision higher up also, and so you'll begin to get cell growth higher up, and that will stop the infection as it's moving upwards. That did not happen with this one. This one just really took off well. So, this plant that I took this one off of, like I told you, I chopped the head off basically. So now it's got 4 shoots coming out from where I chopped this one off. So I'll cut 3 of those off, root those here probably within a couple of weeks, and then I'll train one of them just to be the new leader for the tree, and then plant that out in the yard. So let's see, did I miss anything? Okay, so we've done the incisions. I'll then take the cutting and I have it in the IBA, and I basically I put my IBA solution in just a glass mason jar, and I put it on a little stirring plate too with a little stir in it, so it's kind of swirling around the whole time that I'm doing the cuttings. Then I'll put the cutting in there and put it in there for about 10 seconds. I really kind of want a lot of that to kind of soak into the incisions. And then after I swirl it in the IBA, I then lay them horizontally. If we go back to this slide, this is where I do it all. This is the sink in my laundry room. And then I'll just lay the cuttings horizontally over this. And then I kind of wait for it to more or less dry, just so that as much of that IBA has soaked into the stem as possible. And then that usually just takes a few minutes for them to dry because you've dipped them in 50% ethanol, so it evaporates pretty quickly. And then I'll stick them into little pots of 100% perlite. I only use perlite. Stick them in there kind of tightly, not too tightly. And then I'll just soak them with water from the sink, and then I'll put them under mist. One note that I put down here: if you are going to mix your own, you're going to probably be using your own container. And so I use glass jars, which are clear. IBA does apparently degrade in light over time. I don't know at what rate it does, but just to be cautious, I always keep it inside of I keep it covered with a box just so that it's in the dark at all times, except when I'm doing the cuttings. All right, here's my system of how I do this. So I do not have a misting bench outdoors in a greenhouse. This is an indoor grow tent, like people who want to propagate houseplants, or like, if you want to grow cannabis, and it's illegal, people use it for that purpose too. I think that's actually why they were kind of invented in the first place, was for the illegal growing of cannabis. But anyways, a lot of people use them for houseplants now. And that's why I originally built this indoor grow tent, I was breeding anthuriums, and you can actually see some of them are still sitting in here. But then I was kind of doing trying tissue culture for oaks at the same time, and then decided to just build a misting bench in here. So you can see I just use these clear plastic storage bins you can get anywhere. And then because this is in my laundry room, it's in the basement of the house, it stays a very consistent temperature. I mean, obviously gets colder in the winter, but I'll put a small space heater in here in the winter. So it's generally kind of warmish room temperature like temperatures all the time. It never gets very hot, and I never allow it to get very cold. And so again, it's in the laundry room in my basement, so there's a drain in the floor. So you can see there's little tubes that come out of here. These are the drain tubes, and so that drains into the floor drain. And you can't see it here, but on this rack are misters, just like those little misters that you see like in hot environments where they have outdoor seating or something. At restaurants, they'll have misters going. So the type of misters you can just buy at any hardware store or order online. And then I'm using artificial light, LEDs. That's what these are here. It's pretty bright. it's bright enough that if I'm growing out an oak underneath it, its leaves will look as if they kind of grew in dappled shade. So it's pretty bright, but definitely not the equivalent of full sun. And I'm giving you sort of some data here so you can get a sense of it. So there are 3 of these lights here. They're each 4 foot long. They're each 26 watts of natural white LED, and they're about 12 inches above the top of the plant. So that's kind of enough information if you kind of do the physics and the math there to figure out roughly kind of what the luminosity is where the plants are. But again, like I said, it's pretty bright. And now, if you don't have this set up, you can still do everything that I was just mentioning before, but do it in a normal misting bench in a greenhouse. I'd be really curious if any of you do start trying this, but you're doing it in a normal outdoor greenhouse misting environment, how that works out for you. But I don't have that. I only have one small greenhouse that I have lots of cacti growing in, and so I just don't really have the room to do that. And then, so for the mist, as mentioned here, is the misters turn on for 1 minute every hour, 24 hours a day. So they're kept very moist. And so inside this environment too, it's almost 100% humidity. So they don't have to be misted terribly often. They really don't dry out very, very, very fast. And then here's a close-up down here of, I just use, these pots are, you can see roughly about how big they are, filled 100% perlite. And then, so these are cuttings I stuck just about a little over, about a week ago. And I was using very high concentration fungicide, and you can see most of these look very, very green. They're actually a little bit more brown now. I think I took this picture just maybe only 2 or 3 days after I stuck them. That salt will affect them for up to about 5 days, and then they sort of stabilize. But they're looking really good that I've used the proper amount of fungicide for these. So I'll be excited to see how they turn out. Now, this is the part that I never thought would be very difficult, which is transitioning these into soil. I also have a list here of ones that I have rooted in the past. This does not necessarily mean that they are now growing, because I've probably lost over half. So let me start again. Over half the cuttings that I've got to produce roots, I have lost in the process of getting them to grow in soil. And my thought here is that just as these cells are beginning to grow very rapidly, producing roots, the stem is just, again, very sensitive to infections, most likely, I guess, fungal infections. So a lot of the problem, I think, has been my fault that I'm not being patient. I think the key is to keep the cutting under mist, growing roots in perlite for longer than I'm willing to let it go, because I'm impatient and I want it to start growing in normal soil again. So I think the key is to allow the cutting to grow a pretty substantial root system in perlite under mist. And then once those roots have gotten kind of hard at the base of the stem, I think it probably will be much less likely to get infected. So my protocol that I'm kind of using now is that once I've got a decent root system under mist, I then will take it out, and then I'll just add a little bit of coconut fiber to the perlite, or core fiber. It's written here, COIR, if you're not familiar with it. It's a very fine coconut fiber. And the reason I use that is because the way it's produced, it's pretty sterile. I think there's a lot of heat involved with it. I'm not sure exactly how it's produced, but there's not a lot of mycorrhiza growing in it and things that might cause an infection. So I'll just mix a little bit of core fiber with the perlite, and then move it. I might actually leave it under the mist for a week or two and then move it out of the mist, but keeping it inside the indoor grow tent. I'm definitely not going to expose these to, a harsh environment of a greenhouse in the summer where it's going to be getting direct sun or getting really high temperatures during the day. Not going to do that until I'll really grow these out in the indoor grow tent until I get a flush of growth on it. And then I will be willing to move it to the greenhouse, unless it's the fall cuttings. So the fall cuttings I've taken in the fall that have rooted, I will move them out to the greenhouse because it's cold and humid and not very sunny. So I'm sure they won't be too stressed. All right, I think I mentioned everything there. I think that might have been it. Yeah, so this is actually the crassifolia from here. This is the whole cutting you can see. So this was I think the third flush of growth off of the seedling. This is the flush that just was too floppy to hold up, so I just chopped it off, and this happened in 2 weeks. So very happy about that. So I definitely will be keeping at least one of these once I've rooted several, keeping at least one of these in kind of a coppiced state where I'm just constantly cutting off the growth and keeping it sprouting out from near ground level, because then I'll sort of have a stock plant that will be good for rooting this, for getting a lot more cuttings off of. And then this last picture is McDougallii or McDougallii from Oaxaca. And that's it. So thank you very much. I hope you all give this a try if you're interested, and I definitely want to hear some feedback, and I hope it's positive feedback, that you have a lot of luck with this.
Yeah, wonderful. Thank you so much, Greg. All right, there's a lot of discussion and questions going on in the chat, so I'm going to try my best to go to the top for the first. Well, Brent put in a question about any reason to think this same protocol wouldn't work with deciduous oaks.
So I do not. The only difference there would be taking these in the fall, because then they will be without leaves. But for summertime, I don't see any reason why it would not work. The reason I put that into my title is because I've never tried rooting a deciduous oak. Because I have one deciduous oak in my yard, Havardiana or Havardii, forgetting which one it is, one of those, the shinnery oak. Otherwise, I only grow evergreen oak. So that's really the only experience that I have. But yeah.
Okay, thanks. So I'll just get through the questions in the chat because I think those came first, and then I'll get to you, Roderick, and then Brian, just following the order. So another question was, what's the grade of the perlite that you usually use?
I guess it would be what you call so when I go to purchase perlite, they only have two types, which is what I think of as typical standard perlite, and then these really big chunks of perlite. So it is, I would just call it is the standard size of perlite that you would see that's mixed into potting mixes. Does that help?
Okay. Yeah, and then they put in parentheses just a description of like the best grade of perlite for plant propagation is medium to coarse. This size balances essential moisture retention with optimal airflow to prevent cuttings from rotting. So if you know I guess, if it's medium to coarse perlite?
Probably not. Probably what I use is more medium to fine, I guess. There's definitely is fine particles in it. And that's probably a good point to make. If I can find medium to coarse, that might be a good thing to try. I buy it in these gigantic bags, and it only comes in 2 grades. And the coarser grade is basically like huge pieces of gravel. It's very coarse, but I don't know, it might be worth trying.
Yeah, thank you for the questions. Are the lights on 24 hours?
Good question. Yeah, I forgot to put that in there. No. I typically, during the summer, I mimic the photo period that's outdoors. I have a timer, and so I can adjust it throughout the summer. But then once fall comes along, because I do keep cuttings in there through the winter, I never let the photo period the daylight get less than 12 hours.
All right, thank you. Sorry, have you found a reliable misting timer for controlling the intervals?
Yes, so what I use, it's a little battery. So because my system is in the laundry room, there's a water faucet like for the clothes washer. And so you can buy little battery powered timers that screw onto a faucet. And so that's what I use. And so basically the minimum setting I can do is 1 minute, a 1 minute period. I would really like to do 30 seconds twice an hour, but that's not an option on the timer that I have. But 1 minute every hour seems to work just fine. Oh, and also see vermiculite. I've not ever tried to use vermiculite. The reason I use the perlite that I do is because I buy it in these gigantic bags from the store that kind of sells supplies for people that grow cannabis. And so the big bags are only, it's just big bags perlite. It's super cheap. And I go through so much of it. That's why I've never tried using vermiculite.
All right, thank you. Do you have a success rate for every species?
No. I would say that, I mean, I have a sense of ones that are harder and, and easier, but honestly, I couldn't say my sample, the success rate is so low, for some of them that, and say statistically, I would not be able to, it's not a big enough sample size. But like for the Pinnavinia, the one that I'm such a fan of, I would say I probably get close to 75% success with that one. But there are others that I have had zero success with.
And then the younger the plant, the best to root its cuttings?
Yes. Yep.
Okay. And then I think this is the last question in the chat, and then I'll go to you, Roderick, and then Brian, and then the rest of the chat questions. Are there sterilization concerns if using an outdoor retention pond as the water source?
So that meaning the water source for the mister?
I think this is your
Or
question, Ashlyn. If you're still here, if you want to unmute and clarify.
She put it in the chat. She answered yes.
Oh,
yes.
yes. Okay.
Yeah, I would think that water would probably have some pathogens in it. I would be kind of scared of doing that.
All right. Thank you all for those questions. Okay.
Well, someone's asking about my water, so I'm just using tap water. And unusually, I mean, we are in a city, but our water, ideally, I actually would like to have water that maybe had a little bit of chlorine in it, so it would be a bit cleaner. We, surprisingly, for a big city, we have virtually no chlorine in our water. It actually does come from a reservoir high in the mountains, but it is tested to be, I mean, it's obviously potable and people don't get sick from drinking it, but it does not really have any significant amount of chlorine in it. But it's very clean and very soft water.
All right, thank you. All right, so I'm just kind of going in the order that I think questions came in. So I think those were the ones in the chat that came in first. So then I'll pass it off to you, Roderick, and then Brian.
Thanks, Amy. Greg, that was fantastic. Thank you very much. You can see from the questions it's created a lot of interest. A couple of comments. I was really interested to see you use vodka. But surely for Mexican oaks, you get better results if you use tequila or mezcal. No?
Yeah, I mean, I maybe should try that. It just costs a lot more.
All right. Less facetiously. Well, the comment on evergreen oaks, correct me if I'm wrong, but I think crassifolia is deciduous or semi-evergreen in the wild. And insignis you mentioned as well. I think those can be deciduous in certain conditions. So I think you're already using deciduous oaks, no?
What the insignis and the crassifolia that I have tried cuttings from are evergreen. My insignis in the yard is evergreen.
In your yard, yeah, but I know they're described as semi-evergreen in the wild. So I think you're already dealing with oaks that are not necessarily.
Yeah, I don't see why this method would be any different for deciduous oaks.
Right.
Except for the issue of taking cuttings in the fall.
Right. And my other question was that I've very little experience with cuttings. I'd always been taught with cuttings with a root, you sort of cut it like a whittle stick to expose the cambium. So you're saying you don't do that? That's not a good idea, and it's better to just slice down the stalk?
That's what I have done. Yes. Yeah.
Okay. So
I have seen what you have said.
Yeah.
I've seen that technique, but I just from a point of view of doing it, it's easier to just do that thin incision. Yeah.
Great. Thanks.
Great. Thanks.
Okay. One note: in about 5 minutes, all the students here in high school are about to get out of their class, and they're all going to get on their phones. So if I start to cut out, that's why. Think
Okay.
about it.
Hopefully we can get through the rest of these
Yeah.
questions. Yeah. No worries. Okay, Brian.
I'll try to be quick. I just have three random questions. If you've tried certain things, they're sort of things I've been wanting to try myself. With a mature tree, have you tried giving it a really bad pruning job to get a sort of more juvenile growth growing from a branch as opposed to cutting it down to the ground?
I have not done that mainly because these mature trees I'm getting the cuttings from are not my trees.
Right. Okay. Yep.
Uh, but yeah, so there's a crassifolia that's at Sistus Nursery that is, I think it's probably 20 years old, roughly, is very large, beautiful tree, completely evergreen. And I have gotten it to callus, but no roots though. I mean,
Okay.
another option, which I wish I could have tried this years ago, is, and this is what I've heard people do, is on trees like that, you will graft it. So try to graft it onto something, even if it only the graft works for a few years, that new growth off of the young grafted tree is likely easier to be rooted.
Okay. And then also, have you done anything with like shading? Like maybe use a piece of Velcro to shade a piece of the stem before you cut it off the tree to try to root it. Sometimes that helps with certain woody plants.
Right. I have heard about that. The more elongated it is, the easier. But no, I have not. I have not tried that.
Well, not even just the elongation, but like it's just like a little slip of Velcro and you press it onto the stem. So where you want the roots to form, that is shaded.
Oh,
And sometimes that
oh,
helps depending on
I
the
see.
species.
I have not tried that. No.
Yeah. And then the last thing is have you ever done like a fungicide application when you're potting up the rooted plants?
That would, so getting into that issue of them getting infected after transferring them, that is probably something I need to start trying. Uh, that's just, yeah, I need to think about what to use if I would use that same systemic fungicide. I worry that the roots being so tender that they might get burned by that fungicide. Uh, and yeah, but that
So,
would be a good thing to try.
something I've been curious about, which I haven't tried it, but I know other people use it for rooted woody plant cuttings. It's called Root Shield, and it comes in a granular form, and it's a biofungicide, so it's a beneficial fungus. I guess it just sort of crowds out the stuff you don't want. You can just mix it into the potting mix.
What did you say that was called? BioShield?
Root Shield.
Root Shield.
Yep.
All right, I will check that out. So that would be something to possibly use when transferring out of the mist into more of a soil. Okay,
Right. Yep.
I will try that.
Great.
Great.
Thanks,
Thanks.
Brian. Okay, so I think there were two questions in the chat that came in before the Jesse and Ugo, so I'll do those, and then Jesse and then Ugo. So, actually three, but one was just asking if you have social media, Greg, that people could follow you on to if you post anything about your work, that people could just learn more about. So if you do, and you don't mind sharing that in the chat, someone asked about
Yeah,
that.
so actually, if you're on, so only Instagram is the only place. So if you go to the iOS Instagram page, you should see the post for this webinar that I just gave, and it's in there.
Great. Thank you.
Mhm.
Have you tried this method with any other hard to root woody plants?
A little bit. I've tried it on arbutus. With the Mediterranean species, arachnoid. What? Anyways, what the arbutus species that's native to the Mediterranean? I'm forgetting the species of that. Not unido. I see it. Arachnoides? Is that? Am I? Andrachne. I knew arachnoides didn't sound right. Like that's a spider, right? Yeah, Andrachne. Yes, and yeah. So I'd tried arbutus Andrachne, and again, I did get it to root. I got a flush of growth, but like a lot of those arbutus, getting them established in the soil can be tricky.
Mm.
And I think I tried to do it in the spring, which usually with arbutus and manzanita, I like to plant them in the fall when I move them into the soil outdoors. Uh,
All right. And then
but
there
that's
was
about it. That's about it, though. Yeah.
Yeah, and then have you ever tried no light? People in bonsai extract Quercus with a black bag over method,
I have not. No.
right?
But yeah, all of these other methods that you might have ideas about combining these things together, I think are great experiments, especially if you've got the time and the space to try a lot of these. I mean, I wish I had the space and source of enough cuttings and the time to do a lot of controlled experiments. But I've done a little bit of what I would define as a controlled experiment, you know,
Mhm.
just changing one variable, but not too much.
Okay. Jesse, did your question get answered, or I just want to make sure if you had a question.
No. Thanks, Greg. Just quick question. The how long before the cuttings start flowering? Have you seen that? And then it doesn't look like there's a taproot coming out of the rooted cuttings. Do you have a sense of how they do long term?
So I have a rooted cutting of Quercus pinnavinia in my yard that's about 1 meter tall. It's doing great. So right, they will most likely will not perform or grow one single taproot down, but it doesn't seem to matter to them. And as far as flowering, so again with the Quercus pinnavinia that I've that I'm such a big fan of,
Oh no.
there
Perfect timing.
I gave one house, and so this year it put on. This year would I'm cutting out.
Yeah, maybe just try and turning off your video, Greg. That sometimes helps with the connection.
Okay, all right. What's it saying? Oh, it was a rooted cutting. It was this would have been its third season of growth, and it is flowering. So in 3 years from rooting the cutting, it's flowering.
Okay, great. So yeah, maybe
Male
you'll have
and
acorns
female
soon.
flowers. Yeah, the pinnavinia, the what? So, like I said, the way we kind of determined this was Quercus pinnavinia was it produced female flowers, but it was only just recently that one produced male flowers. So there's not been any acorns because there's only been female flowers coming on these trees. But
All
starting
right.
starting to see some male flowers too.
Great.
All
Thank
right.
you.
Thank you. And I just want to say, just so we can get through all the rest of the questions, I think we're going to cut off questions, but we'll get through the rest of the ones people asked in the chat, and we'll go to Ugo, you first. But yeah, Ugo, go ahead.
My question was already answered. It was about the flowering.
Okay, great. Perfect.
Yeah, and also if
Thank
you
you.
have any other questions, just send me questions on Instagram. Just message me on Instagram, and I can also like give you my email too, and we communicate that way.
Awesome. Thank you so much. Have you tried air layering oaks?
I have not. No.
All right. And then do you track the ambient humidity and temperature?
Well, like I said, I the only time I do anything about the temperature is once winter comes and it starts to get cold in the basement. I will put a little space heater in there just to keep it, what you would kind of consider room temperature. And as far as the humidity goes, I have a dehumidifier in the room where the grow tent is, but in the grow tent, it's kept closed with a few vents on it. So I would say it probably never gets below 80% humidity in the area where the misting is happening. Sometimes it's probably getting up close to 100% humidity.
All right. Well, thank you all so much for the questions and discussion. And thank you, Greg, for your responses. And again, thank you for your discussion and questions for Ugo's presentation and your responses as well, Ugo. And a huge thank you again to both of our presenters for sharing their really incredible and interesting work with oaks. So thank you so much again for taking the time to put this together and to share with everyone. Much appreciated. I think now we're have a few minutes. I don't know if Roger, you have any final thoughts or questions for the group while we're still on.
Amy, one thing I'd like to do is share the results of the poll. Are we able to do that? It's really interesting to see. We had about, I think it was 45% non-iOS members, which is great. And if you like what you saw, you want to be more involved with the iOS, by all means do join. There's information on the website on how to join, or just get in touch with us through the website and we'll assist you with that. But nothing else from me. I think we've had two great presentations, so thank you, Hugo and Greg, for the time in preparing this and for presenting. Really interesting stuff.
Yes,
All
thank
right.
you.
Well, thank you very much.
Great.
Yes, and I put the link to the page on how to join the iOS for anyone interested. And thank you all so much for attending today's webinar and hope to see you again in the future. So have a great rest of your day, everyone.
All right. You too.
Thank you.
Thank you, Amy.